Excellent. So we are joined today by Dr. Wyatt Yu, who is a professor of structural biology at Newcastle University and who will be moderating this session. Dr. Yu, over to you.
Thanks, Calliope. Good morning, good afternoon, everybody. As Calliope said, I extend the welcome to this lightning session. We have today 5 excellent talks from 7 very, very excellent researchers at the forefront of therapeutic discovery, particularly in the direction of substrate reduction and polyglucosan body reduction. I will just add to Calliope's point that if you have any questions, please put into the comment box, or directly message me in the comment box, or raise your virtual hand at the moderating session. So without further ado, I'd like to introduce the first talk presented by 2 speakers, Bersh Minassian and Sylvia Nitschke from University of Texas Southwestern Medical Center. And Bersh and Sylvie will be presenting their work on GYS1 targeting antisense oligonucleotide and siRNA. So look forward to this talk. We'll pass the floor to you, Bersh and Sylvie.
Thank you, Wyatt. Thank you, Calliope, and all the organizers. Thank you, Aaron. I will not speak long. I'll just say that polyglucosan bodies are made of polyglucosans, and polyglucosans are characterized by chains of glycogen that are too long, which leads to their precipitation and accumulation into the polyglucosan bodies that cause disease. So it's not rocket science to think that if we could prevent this over elongation of glycogen chains, we should be able to prevent formation of abnormal glycogen and therefore polyglucosan bodies. There are many ways to downregulate an enzyme. You'll hear about them in Sylvie's talk, but in the rest of the session as well. Just to say that we've kind of pushed strongest forward with the antisense oligos because they cross they're accessible to the brain through delivery by lumbar puncture, and because there are clear examples of it crossing the FDA threshold and going into the clinic. So clinic being a priority, we've pushed for strongest on that avenue. I will not go further. I saw Sylvie's lecture. It's really well done. So I'll introduce Dr. Nitschke, who's been a colleague working in the lab for a number of years. Yeah, I'll just let her present this work. Sylvie, please take it away.
I unmuted myself successfully. Thank you, Bersh. Hello, everyone. I'm really excited to be here. So let me jump right in. Bersh already introduced a little bit, and my next slide, hopefully, doesn't move forward.
How do we advance slides, Aaron?
Oh, could you try clicking on it, or also using your arrow? Yeah, great. Thank you.
Yeah, so let me say a few words about the rationale behind. Bersh already mentioned it a little bit. Normally, we have soluble glycogen, which is nicely structured and synthesized by 2 enzymes that act in concert, which are glycogen synthase, which is responsible for chain elongation, and then the glycogen branching enzymes, GBE, that introduces the branch points. And a well-balanced action of these 2 produces soluble glycogen. In disease, in polyglucosan body disease, like APBD or Lafora disease, abnormal glycogen is formed, which we call polyglucosan, which becomes insoluble over time. Characteristics of these polyglucosans are that they are overlong chains, glucan chains, and less branch points, which are responsible for making those molecules insoluble in the end. So basically, the rationale behind our therapeutic strategies to prevent polyglucosan bodies are to attack glycogen synthase, basically to reduce glycogen synthase to prevent those long chains and this imbalance between chain elongation and branching. proof of principle studies were done using a genetic model. Glycogen synthase knockouts were bred with Lafora disease mouse models, in this case Lafora knockout, and with APBD mouse models, GBE deficient mice. On the left side, you see the magenta dots are the polyglucosan bodies, which we are able to see when we do PASD staining. And then on the right side, we see the mice bred with glycogen synthase knockout, and you see the polyglucosan bodies were completely prevented in the full knockout and very well reduced in the monoallelic knockout, so the crossed with heterozygous GYS. So this is a down-regulating glycogen synthase is a very valuable and promising therapeutic strategy. Over the years, we and others have published a number of papers showing exactly that not only the glycogen synthase knockout model was used, but also other ways of down-regulating GYS, for example, by reducing regulatory proteins that are basically indirect activators of GYS, so such as PTG or R6, or using, and this is what I'm going to talk about today, using approaches to silence GYS gene expression. And this is done by antisense oligonucleotides and artificial microRNA. I'll start off with talking about the antisense oligonucleotides. We published a paper focusing on Lafora disease only last year, but I'm gonna also present you a set of unpublished data about APBD, which are also looking very promising. So how do antisense oligonucleotides work? Here, the antisense drug in red would bind via Watson-Crick base pairing to the mRNA of interest, in our case, glycogen synthase 1. And upon that binding, RNase H, an enzyme that is able to degrade the mRNA, will be recruited, and then the mRNA will be degraded, and no more protein translation will happen. On this ASO project, we worked together with Ionis Pharmaceuticals, and they actually developed a lot of potential ASOs, basically covering the whole mouse GYS1 gene, as you can see here, and they tested those ASOs in vitro, and yeah, you can see a few bars show substantial reduction in mRNA and GYS mRNA expression, and we basically selected the one in black here. You can also see a nice dose-response relationship that could be achieved. So, and then we went ahead into mice to test, are we actually able in vivo to downregulate GYS mRNA expression? And we were able to. In different regions of the CNS, cortex, hippocampus, and spinal cord, we saw significant downregulation of gene expression. So now we went into Lafora disease mouse models. Here I will show you the Laforan deficient mouse model results. The ASO was injected at 1 month and 2 months, and then tissue was collected at 3 months. We had two control conditions, PBS injected mice and control ASO, which is a non-targeting or mismatch ASO that won't bind. And of course, the GYS targeting ASO, which led to a pronounced downregulation of mRNA levels. Also, on the protein front, we saw a substantial reduction. And yeah, we could actually see nice prevention of glycogen accumulation. Basically, the amount of glycogen that you see in the controls are due to the accumulated insoluble glycogen, the polyglucosan glycogen. And with the ASO treatment, we basically have wild-type levels. Yeah, also here, the PASD images show that the ASO was able to strongly prevent polyglucosan body formation. In Lafora disease, we call them Lafora bodies. Yeah, similar results were obtained with myelin deficient mice. Now I will move on to a slightly different trial where we wanted to analyze neuroinflammation. At 3 months of age, our Lafora disease mouse models don't show that much neuroinflammation yet. Therefore, we went ahead and had a longer trial where we actually can analyze neuroinflammation as well. Treating or ISCV injecting at 3 months, 6 months, and 9 months, and collecting the tissue at 12 months, but also collecting tissue at 3 months, just as a baseline, because at 3 months we already have a few LaFora bodies. Let's just look at the PASD staining here. For time's sake, we could achieve a strong hold in disease progression with the ASO, with the GIST targeting ASO. And what about neuroinflammation? When looking at GFAP, which is a marker of neuroinflammation, we also could see strong prevention of neuroinflammation using this ASO. So now I'm going to jump to the APBD part, which I told you in the beginning about. So here, the setup was slightly different. We already learned from the LaFora disease studies that we can actually strongly prevent disease progression, but we cannot reverse it. And since the APBD mouse model is a little bit more affected and accumulates polyglucosan bodies a little earlier, we even started a little bit earlier with the first injection. We injected neonates then at 1 and 2 months again, and collected tissue at 3 months. And again, we achieved downregulation of mRNA levels. Glycogen accumulation was strongly prevented, and also, as you can see on the PASD stainings, polyglucosan bodies were strongly prevented using this ASO. So this ASO therapy is very promising. This preclinical study led Ionis to produce actually the human. This was only the mouse ASO targeting the mouse gene, but Ionis developed the human ASO, and it would technically be ready to go into a clinical trial. Now I'm gonna move on to artificial microRNAs. This project was published this year, and there's a slight difference here. The oligonucleotides are not directly injected here. Viral delivery was used. Basically, the artificial microRNA expression cassette is packaged into an AAV9 virus. Here you see the scheme of the miRNA, and what is really important is the blue part, the stem region, that is has the sequence that is responsible for targeting the glycogen synthase mRNA. We used a bicistronic expression system, basically under the CBH promoter, or CBH promoter drives expression of both the miRNA and GFP. And here you see an example of miRNA or AAV injected mice and PBS injected mice. And you see this is GFP expression that we achieved a very good spread and nice GFP expression reflecting miRNA presence. Let me start with the LaFora disease mice again. Again, LaForin deficient mouse model. Here, it was only 1 injection that was necessary, and this was done at postnatal day 2. And then the tissue was collected at 3 months. We achieved about 15% downregulation of mRNA levels, but as you can see on protein level, the effect was much stronger. We achieved about a 50% reduction in protein levels. And this led to a strong prevention of insoluble glycogen formation. This is only, it's not the total glycogen, this is the insoluble glycogen only that we determined here. More than 50% or about 50% reduction. And also on the polyglucosan body or LaFora body formation level, we could strongly prevent their formation. As I mentioned already earlier, at 3 months, neuroinflammation is not yet that pronounced in LaFora disease mice. We only have 1 neuroinflammation marker that already shows an increase at this age. So I show you this example here, and we see it's not a complete rescue, but it's definitely a significant rescue that we could achieve using the miRNA targeting GYS. Yeah, again, for myelin deficient mice, we obtained similar results. I'm not going to show this today. Now, the APBD mouse model, it's the same experimental setup that was used. Again, about 15% reduction in mRNA levels could be achieved. And again, the protein levels were reduced a little bit stronger than the mRNA levels, which is great for us. You can probably also nicely appreciate that the protein levels really correlate with the GFP signal that we could see. Yeah, so insoluble glycogen as well as polyglucosan bodies were also significantly reduced using this approach. And here I show you the same neuroinflammation marker, CXCL10, which shows a much stronger upregulation in the PBS group in APBD mice, because I told you they are more affected at the same age. And yeah, a significant prevention of neuroinflammation with the miRNA. Yeah, to conclude, as I already told you in the beginning, we think downregulating GYS is a very promising therapeutic strategy for polyglucosan body diseases such as Lafora disease and APBD. There are, of course, so basically the preclinical studies in mice are very, very promising. Now it's about translating this into the clinic. And there are, of course, some hurdles for viral delivery. It would be to achieve good transduction efficiency throughout the whole brain. And for ASOs, it would be, as Birch mentioned, again, to do repeated lumbar punctures, but this is something that could be done. But I will hand over now to Birch.
Thank you, Sylvie. We wanted to just conclude by a word. You may ask, why aren't we already in clinical trial, especially with the ASO, because that has a clear path forward. The reason is we need $10 million to run a clinical trial, and there are active negotiations going on to try to get some industry support so that we can run that clinical trial in Lafora disease. To run the clinical trial in APBD is also possible. That will need also that amount of money. It seems that the Lafora disease will go first because that disease is very devastating and rapidly progressing, and proof of efficacy can be obtained easier than with APBD, where it's harder. It'll take longer period of time to prove that a drug that prevents progression worked. But nonetheless, I mean, the study can be done in either disease or both diseases. If it is done in one disease and proves safe and efficacious, I think it can be used in the other disease kind of off-label, if you like. We can talk about this more later today and maybe tomorrow. But right now, the plans are to try to go into clinical trial ASAP with Lafora disease, and negotiations are ongoing to try to secure the funds we need to actually run the human clinical trial. We will stop here, and hopefully we're on time. And Wyatt?
Thank you very much, Sylvie. Thank you very much, Birch. We can reserve some time for comments and questions at the end of the section. So I would then introduce the second speaker and the second talk by Julie Ullman from Maze Therapeutics. So Julie will be speaking to us about developing small molecule inhibitor for GYS1 as substrate reduction therapy for Pompe disease. Julie?
Yeah, thank you, Wyatt. Let me pull my screen up. Let's see. Yep. Okay. Whoop. And okay, am I in presenter mode here?
Yes, that looks great.
Okay, great. Well, that Burj and Sylvia, you couldn't have given me a better introduction. Thank you very much. I think what's, echoing some of the sentiment, what's really exciting about development right now for these glycogen storage disorders is that there's multiple modalities being developed and being ready to be tested in the clinic. And I think folks who have worked in clinical development know that, everyone goes into it with the highest hopes and aspirations, and then you get into human patients and you see what you see. And I think the best thing for patients right now is that we are in a position to be testing different modalities and see how well they work and how well they actually fit into the clinical maintenance of the disease for patients. And so I'm very excited today. Let me just set my timer, apologies. Very excited today to be representing Maze Therapeutics and to be telling everyone a little bit about our new investigational drug, MZE-001, which is a small molecule, small compound inhibitor for glycogen synthase 1 that we are currently developing as a substrate reduction therapy for Pompe disease. Oops. So, again, thank you for the introduction of glycogen storage disorders and specifically the polyglucosan body disease. Glycogen accumulation and glycogen storage is a feature of many different diseases. And these glycogen storage disorders collectively sort of run the gamut of enzymes across the glycogen biosynthetic pathway. The enzyme here, muscle glycogen synthase 1, is considered to be the rate-limiting enzyme in this glycogen biosynthetic pathway. And it's been long hypothesized and demonstrated in several different therapeutic modalities and in lots of different preclinical models to be beneficial targeting GIS1 for a multitude of glycogen storage diseases. We at Maze see potential for targeting GIS1 in several different glycogen storage disorders, including Pompe disease, our first indication that we are focused on. But it potentially has a real benefit in several other patient indications, including adult polyglucosan body disease, Lafora, McArdle's, and Cory disease. So Pompe disease is somewhat unique amongst the glycogen storage disorders. So I want to spend some time to just give you the background of what makes this disease unique, especially relative to the adult polyglucosan disease. It's primarily a muscle dysfunction, and in muscles, muscles utilize glycogen very heavily for an energy resource. And so glycogen synthesis in a healthy muscle cell basically starts with glucose entering into the cell. It's then converted into glycogen through a series of enzyme steps. Again, the rate-limiting enzyme is GIS1. This glycogen that's produced in the cell in the cytosol is used and recycled and turned over for glucose monomers, but some portion of that glycogen that's synthesized ultimately traffics or transports to the lysosomal compartment, where it is turned over and recycled by the lysosomal enzyme acid alpha glucosidase, or GAA. And it's loss of function mutations in that enzyme GAA that are causal for Pompe disease. And this initial insult of accumulation in the lysosomal compartment triggers a multitude of other sort of pathway level dysfunctions of specifically and most severely dysfunctions across the autophagal lysosomal pathway, sort of a trafficking pathway in the cell that triggers other dyshomeostasis in the cell, including profound metabolic dysfunction. And so current therapy for Pompe disease focuses on replacing that GAA enzyme, and that's called enzyme replacement therapy. And this enzyme replacement therapy works very, very well for the tissues and the cells that receive the therapy. However, with any sort of large molecule biologic, there are challenges with biodistribution across the body and across the different cell types that need it. And for Pompe disease specifically, there's a limitation of biodistribution to the skeletal muscles that really need it most in the Pompe patients. And so while patients do benefit, especially in cardiac tissue, which is a significant problem for the most severe patients, the amount of enzyme that gets to the skeletal muscle is limiting. And as a result, patients continue to progress in their disease course and ultimately suffer from diaphragmatic failure and respiratory failure over time. So there's a really profound, significant unmet need in Pompe patients, and we believe that substrate reduction therapy could be a very apt therapeutic strategy to address that unmet need in the patients. And, like Burj said, it's actually a very simple concept. You just reduce the amount of glycogen that's made, and if we can reduce safely at a level that's well tolerated, the amount of glycogen that's made in the cells in the target tissues that need it, then we can reduce the amount of flux that's going into the lysosome and accumulating and causing all of these pathway dysfunctions. So at a simple level, that's the concept. And then of course, executing this and then understanding safety and tolerability in humans is another kind of challenge in and of itself. So what I hope to show you today is that we've developed a series of MACE small molecule inhibitors for GIS1 that are potent and selective in inhibiting glycogen synthase activity in preclinical models. Overall, GIS1 inhibition was generally well tolerated in the preclinical studies. We've demonstrated that treatment of Pompe mice with substrate reduction monotherapy significantly reduces glycogen stores, and that importantly for Pompe, combination therapy of substrate reduction therapy with enzyme replacement therapy normalized tissue glycogen and reduced markers of cellular dysfunction in Pompe mouse skeletal muscle within 12 weeks. And finally, that these preclinical studies support evaluation of this novel substrate reduction therapy, small molecule oral pill in Pompe disease patients. So to begin, we started this project several years ago and relied heavily on recombinant protein. So this is recombinant human GIS1 and GIS2. We've developed as well some cryo-EM structures of human GIS1 and GIS2 in combination with glycogenin to look at compound binding. But these initial studies looking at inhibition of recombinant activity were very informative to help us select compounds that had the right chemical properties. And specifically, the properties that we were looking for was selective potent inhibition of GIS1 over GIS2. So GIS1, glycogen synthase 1, is really ubiquitously expressed all across the body, except for in the liver. The liver exclusive isoform of glycogen synthase is GIS2. And in Pompe disease, there is not a significant burden on liver function or liver glycogen accumulation. And so by not targeting GIS2, we're able to preserve the whole body glucose homeostasis mechanisms, whole body glucose handling mechanisms that the liver is really responsible for. And so these data, the in vitro recombinant data, show that across multiple species, we have targeted select inhibition of the compounds against human, mouse, and rat GIS1, and we don't see inhibition in the GIS2 isoforms. And these in vitro studies translate really well to all the different cell culture models that we've studied. And here on the right, I'm just showing you the data from one such cell model, Pompe patient fibroblasts, where we have a potent dose-dependent inhibition in these cells as well. So then the next question really becomes, how do we evaluate this in live animals? And so to do that, we developed in collaboration with the UK group and Gentry's lab, an assay where we evaluated de novo glycogen synthesis in live animals. And so what we do is a short fast period with and without drug. And then we apply a bolus of heavy labeled glucose and then allow that heavy labeled glucose to become incorporated into newly synthesized glycogen over a 4-hour period before euthanizing the animals, collecting the tissues, and then ultimately extracting the glycogen and analyzing it with very high sensitive liquid chromatography mass spec. And so the data that we see here when we apply this analysis and look at tissues that are most relevant for Pompe disease we see nice dose-dependent inhibition of acute glycogen synthesis in the gastrocnemius, the diaphragm, 2 examples of skeletal muscle that are really impacted in Pompe disease. And importantly, we also see inhibition in the cardiac muscle as well. And what's really key here is that we see this targeted GIS1 and muscle-specific inhibition of glycogen synthesis, and we do not see glycogen synthesis inhibition in the liver. So this is specifically designed for this. It's also important to note that these compounds and the MZE001 are clinical candidate in all of the preclinical studies that we've evaluated for tissue biodistribution. We do not see significant exposure in the CNS, in the central nervous system, but we do see really potent and high distribution across all of the periphery, across all of the target tissues that we design the molecules to do. And so then the next question becomes, how does this acute synthesis inhibition translate to impacting steady state equilibrium levels of glycogen in key skeletal muscles in the Pompe mice? The study we designed to evaluate this is first a monotherapy study. Here is the data from wild type mice. So these are animals fed with SRT compound, substrate reduction therapy compound formulated in chow and delivered at an exposure level that reaches the kind of steady state glycogen levels that we believe are clinically relevant for the clinic in terms of targeting healthy glycogen. And so what we see here is a 70 to 80% reduction in total amount of glycogen stores that is maintained from 4 to 14 weeks. And importantly, when we now evaluate the glycogen levels in the Pompe mice, which have, upwards of 10-fold accumulated levels of glycogen within 3 to 5 months of birth, we see a very significant reduction here in total glycogen stores of 40% by just a month. And then after 14 weeks, we see an even greater significant reduction in glycogen in the Pompe model mice, consistent with, the sort of best case ERT that's in the preclinical literature. So this is, really exciting and really important for these Pompe mice, which lack any and all active GAA. And we wanted to understand the kinetics of this at a little higher resolution so that we could evaluate sort of what our next step experiments would look like. And to do that, we again relied on this de novo glycogen synthesis assay. Here's the data recapitulating published data from the literature showing that synthase activity is elevated in the Pompe mice. And when we now look at a pulse chase version of this experiment where we pulse in the 13C6 glucose and look at the kinetics over time, we see, more or less what we expected to see, but there's 2 points that I'd like to raise here. One, the wild type clears the newly synthesized glycogen in skeletal muscle tissue very, very quickly. It has a half-life of 16 hours or so. And this is in stark contrast to the Pompe, where the half-life is, well over a week. But there is a significant amount of clearance over time, independent of GAA. So there is a mechanism in live animals that is allowing for, expelling this accumulated glycogen. And people have hypothesized that this is autophagic lysosomal exocytosis. And that could be a mechanism relevant for other glycogen storage disorders that accumulate these bodies within the cytosol. So next, we wanted to then sort of speed up the process for substrate reduction therapy. And we did that through a combination study where we combined substrate reduction therapy with enzyme replacement therapy. So this is a study design demonstrating, comparison of the wild type with the Pompe mice. We formulated the SRT in chow. We're comparing that to enzyme replacement therapy standard of care. This is the approved drug, Lumizyme, at 20 mg per kg biweekly, and comparing that with the two therapies combined for 3 months. And these are in animals that already show significant accumulation and significant deficits. So this is really the key data for this presentation and really for the program, honestly. So what we see here in the gastrocnemius, to walk you through this, is again that accumulated glycogen in the vehicle Pompe versus the wild type. ERT, the approved drug, has a modest effect in the glycogen stores after 3 months. Substrate reduction therapy is sort of on the equivalent level of ERT in this tissue bed. But what's really exciting and what was really thrilling for us to see, at least in this preclinical preclinical model was that we could achieve, statistically normalized levels of glycogen with a combination therapy in a relatively short period of time in these animals that are, very severe in terms of their glycogen burden. And we see this not only in the gastrocnemius, but we see it in basically all the other skeletal muscle tissues that we've evaluated, including this key muscle, the diaphragm. And importantly, while the ERT very successfully addresses the cardiac glycogen accumulation, we do also see significant impact on cardiac glycogen accumulation with SRT alone. And so next we wanted to understand, how does this change in glycogen impact the disease pathway pathology that we know is really important for Pompe? And to understand this, we've looked at a whole series of different things. This is a very small piece that I'm showing you here with LAMP1 and P62 providing biomarkers or surrogate biomarkers for lysosomal dysfunction in LAMP1 and autophagosomal block for P62, both of which are accumulated in the Pompe, and both of which are significantly impacted by this combination therapy. Suggesting that we are, not only moving the glycogen stores, but we're beginning to really move the needle on the pathway dysfunction that's driving the muscle atrophy over time in Pompe disease. And so last but not least here, looking at the time, the last data I wanted to share with you is we've done a good amount of work as well, investigating the utility of some clinically relevant biomarkers. Urinary GLUC4 is a glycogen breakdown product that's found in both the plasma and the urine. It's a marker for multiple glycogen storage disorders and is a key clinical biomarker for Pompe disease. This biomarker generally reflects what we see in the muscle bed. And again, the additive combination therapy has a really robust effect in this. And then this PBMC glycogen, so PBMCs are a peripheral blood mononuclear cell. And so in this assay, we're able to investigate basically the plasma exposures of drug as a consequence of, or how much that affects the PBMC glycogen stores over time. And we see really nice specific target engagement of our drug and our inhibitor in this cell type. And so we're excited to be exploring both of these biomarkers and the effect of MZE01 substrate reduction therapy on patient glycogen and these fluid biomarkers. So in conclusion, and I know I'm going over about a minute here, but so what I hope to have shown you today and what we're very excited about is that our investigational clinical candidate, MZE01, is a small molecule inhibitor of glycogen synthase 1. It has good distribution of drug to muscle tissues with potent and selective inhibition of GIS1. It's been generally well tolerated in preclinical studies in mice, rats, and dogs. Excuse me, that substrate reduction monotherapy significantly reduces glycogen storage in Pompe model mice, and that preclinical data suggests that there's a possible additive benefit of SRT in combination with ERT. And lastly, I'm very excited to report that we are, in the middle of our phase 1 studies investigating safety, tolerability, exposure in healthy humans. And we are excited to be in the planning phases for our phase 2 Pompe patient studies expected next year. So with that, I'll conclude by acknowledging and thanking the entire amazing team here at Maze Therapeutics, and extending a very warm thank you to all of our collaborators, including the team at the University of Kentucky, our collaborators at the University of Indiana, and Yale University as well. And with that, I thank you for your time, and apologies for going over by a minute or so here. So thank you.
No worries, Julie. Thanks a lot for the presentation. That's great. So again, we will reserve comments on the end, and I can see already some active discussion of the past talks. That's great. So the next talk will be presented by Orca Long and Alexander Lossos from Hadassah Hebrew University Medical Center, and Alexander will present small molecule compounds regarding pharmacological approaches to treat APBD.
So first, I just wanted to present the approach, the general approach, and it's developed development of pharmacological therapies by the understanding of the mechanism of action. First, see the phenotype at the cellular level, and then we zoom out to understand the mechanism of action, showing autophagy induction. Then we see the histopathology, and then we move on, or we zoom out to see the effect in vivo. So the basic approaches for the reduction of polyglucosans are actually two. As you know, polyglucosans are elongated by Gs and are branched by glycogen branching enzymes. So whatever reduces this ratio of Gs to Gb activity can tilt the balance between polyglucosans and glycogen towards glycogen. This is one approach, and the other approach is just direct degradation of polyglucosans, which could be an advantage because it can tackle pre-made polyglucosans, which the changing of de novo synthesis cannot. So our approach actually was initiated by first setting out a high throughput assay, in which the readout was simply stained by periodic acid sheaf with the degradation of glycogen by diastase, and we just looked at quantified the fluorescence by in high throughput format, and we looked for compounds which can reduce the fluorescence of the polyglucosans. Then we screened the library, we found several, and then we screened them in silico using different algorithms so that they can be preferred and not toxic to the cells. So this was an in silico prediction. We confirmed the safety. I'm focusing on one of these compounds that you find in the screen. It was proven to be safe, as you can see, caused no lesions in several tissues, and also didn't change the effect on the mouse weight, and the Irwin tests were satisfactory. And then we just applied it to mice. So when we saw several efficacious effects, increase in the lifespan, this effect was actually quite important, improvement of the yes, so improvement of the extension reflex, which correlates with the pyramidal signs that are observed in patients, and therefore is very important. The extension reflex is the opening of the pores when the mouse is hanged from its tail. And we saw improvement in normal parameters of motor field and gait. All these motor behavioral parameters were improved. Histopathologically, we saw reduction of polyglucosans in the liver, the cortex, the heart, and peripheral nerve. And interestingly enough, not in the muscle, which, as you know, is poorly perfused tissue. And glycogen was affected similarly, was also reduced in these tissues, not in muscle. And this correlated with the pharmacokinetics of the compound. Okay, 144-DG11, it's correct. And as you can see, it accumulates very quickly in the serum. Of course, it drops. But I wanted to draw your attention to the fact that in the muscle, it doesn't build up almost at all, whereas it does build up in the brain, as opposed to glycol, a previous molecule we tested, which did not reach the brain.
Dr. Kaklon, your audio is cutting out a little bit. I think it might be your microphone or how close you are to it. Oh, the slides look great. Just sometimes we can't hear you. If you
Okay.
stay close to the microphone and speak a little louder, that will help. Thank you.
Yeah, it's a built-in. Okay. So the next thing we looked at was actually quite important, and this is metabolic cages, or in vivo metabolism. So this is actually the first time it was done for glycogen storage disorder, and what we did was just measuring several metabolic activities in this advanced system. And what I wanted to point out is that we saw an increase in the carbohydrate metabolism. and this is actually quite important. If I take you back to Biochemistry 101, the respiratory quotient, which was increased by the compound, is actually the ratio of CO2 released to oxygen consumed. So as you remember, if carbohydrates are metabolized and they go through the Krebs cycle, this generates CO2. Whereas if you use fatty acids or amino acid oxidation, you can bypass the citric acid cycle, the Krebs cycle. And therefore, the higher the respiratory quotient is, the more the cells use this pathway. So the more they degrade or metabolize carbohydrate at the expense of fats. And this is actually what we observed: increased carbohydrate metabolism and increased respiratory quotient. And it didn't have any adverse effects on the biochemical blood panel. Okay, while it corrected the hypoglycemia and reduced the hypertriglyceridemia, it didn't affect the liver function or muscle. This was another component of it. Now, is there levels of polyglucosan in patient blood very variable? Okay, we wanted to design a test in which we'll have a uniform effect of the compound. We actually designed glycogen burden condition, which means starving the cells for glucose for 40 hours, and then replenishing the glucose, so the uptake of glucose and the buildup of glycogen is induced, as you can see here. And under these conditions, we did see a uniform effect of the drug, and also you can see that the several cellular parameters that we quantified, okay, by imaging, organellar features, different organellar features, about 45 of them are shown here. So all these effects were actually emphasized by inducing glycogen burden, which means that the glycogen burden actually makes the cells more liable or more sensitive to this treatment, which targets glycogen overdose. So as I told you before, it increased the glycolytic. I mean, not only does it degrade glycogen, it also increased the ratio of glycolytic ATP to mitochondrial ATP production, which means that the glucose, which is released from the glycogen, is actually can be utilized for energy production. Okay, so this is an efficacious degradation of glycogen. So I'll just go over this, we'll skip this. But what I wanted to show is that if we looked at when we analyzed the mode of action bioenergetically, we saw that it actually increased electron transport in the ETC, in the electron transport chain of the mitochondria. And this can be shown by increasing both the membrane potential and the basal respiration coupling efficiency. So it means that you have more energy substrate for the mitochondria as a result of the enhanced glycolysis, more pyruvic acid. So to find the mechanism of action, you need to find the target. And for that, actually, we collaborated with the French company Innoviem, and they have a very nice, very, very sensitive, the most sensitive we know, platform for detecting the heteroassembly which interacts with your test compound. And these are just liquid crystals that we show that are in the naturalized, okay, of the cells, so that they are built only when you add this compound. So they don't bind to one of the vitamins or another small molecule in the lysate. And we found the heteroassembly, which actually interacts. So if it interacts with one, unmute. Okay, can you hear me?
Yes, we can hear you now.
That's great. We do have problem earlier with the sound. If you can.
Yes, I mean it's a built-in mic. So
I see.
Do interrupt me if something is not clear, if it's urgent. So we just continue from where it was disrupted. So we found LAMP1 as the target of compound A, 144G11, and then we confirmed it with SPR, and we saw something interesting. We saw that there is binding, but the binding is pH dependent. So the binding is only in the acidic pH of the lysosomes. However, while specific, the Km was too high, and we actually noticed that this is as you know, Km is actually a K off divided by K on, a quotient K off and K on, and then we saw that the K on was very slow to what we usually observe in SPR, in plasmon resonance. So we thought probably it's the heavy glycosylation tree on LAMP1. So we chemically deglycosylated, and then we saw actually that the AD is normal, 52. And actually, we think that you can see here the binding, that these balls are the glycosylation trees. So you can see, sorry, the of compound A to LAMP1. We think that it disrupts actually LAMP1, it dilutes LAMP1 in the lysosomal membrane, and therefore actually allows for LAMP2 to interact with the autophagosomes. And you can see the treatment actually led to reduction in lysosomal glycogen buildup, and as I said, there is probably dilution of LAMP1 on the membrane, and which enables LAMP2 to interact with the autophagosome, as actually was documented. So we saw that that the compound increased autophagic flux both in cells and in mice. So let's see, 3 and 62 substrate. And we noticed something interesting, that, as I said before, downregulation of LAMP1 is equivalent to the compound. So it actually enhances the effect. If we knock down LAMP1, we also see an increase in autophagic flux, which is enhanced by the compound. Now, what was interesting is that in LAMP1 knockdown cells, the compound actually increased both total and respiratory ATP production, whereas in starved LAMP1 cells, where autophagy is of course induced, it mainly increased the glyco-ATP, the ATP which is derived from glycolysis. As I said before, which is a product of the glycogen degradation. We saw an increase in lysosomal acidification, okay? Again, both by LAMP1 knockdown, which is enhanced by the compound. And we saw that actually other positive effects on lysosomes, such as reduction in their size and an increase in the mitochondrial potential. So there's more substrate, for more respiratory substrate. I'll just go quickly over. We did a proteomics analysis, and we saw indeed induction of lysosomal function associated pathways and of OXPHOS pathways. And so this summarizes our approach. Increasing autophagic flux enhances lysosomal degradation of glycogen or polyglucosans. Now, so now where are we now? So the compound 144 DG11 was licensed to a new Israeli startup company called GHF, Golden Heart Flower. GHF is currently conducting due diligence and is also investing in expanding 144 as the DG1 as an autophagy inducer, safe autophagy inducers to other storage disease disorders, as you can anticipate. So, as Dr. Lossos mentioned, we now have a compassionate treatment in Hadassah. This is the drug that is used, of course, is not injected in DMSO solution. It is applied as a tailored nanoformulation, okay, in which the bioactive is actually on the surface of a tailored specific nano droplet designed by LDS and Western Nissim Gauti. So this just shows you the advantage of this. As you can see, so that's it. We showed, let me summarize, we showed the proof of concept in patient-derived cells, which is also an advantage. So it brings us closer to the translation. It's the first time that we offer a universal solution for storage disorder by inducing autolysosomal degradation. Another innovation is the first time there's a whole animal level metabolic characterization of a glycogen storage disorder. And we found a target and biological mode of action of our drug. Let me just last and not least acknowledge the contribution of my collaborator Miguel Val in Tel Aviv University, all these collaborators, Nissim Gatti and LDS for the nanoformulation, of course, of mice and health and support. Sorry, of course, Alex Lossos in the clinical and GHF, the newly formed startup company, which supports us. Thank you very much for listening, and sorry about the disruption.
Oh, thank you so much. I'm glad we get there in the end and to hear the exciting part. Yeah, that's great. We will move on to the latter part of the session. This will be a talk by Matthew Gentry from University of Kentucky. And Matt will present his research on the delivery of antibody enzyme fusions for the treatment of glycogen storage diseases. Thank you for putting the slides on. From the Matt, all yours.
Look okay there, the slides?
Yep, that's right.
Yeah, I just want to say again, thanks to the organizers, the APDF Foundation for this opportunity to share our work and to fellow presenters for setting up the talk so well. A lot of exciting work in this space, and I'll tell you a little bit about what we're doing here in my lab at University of Kentucky. So you heard a little bit about Lafor's disease. It's a really horrible, fatal autosomal recessive disorder. These children develop normally until about age 12 when they have horrendous epileptic episodes. They also undergo childhood dementia. One other aspect are myoclonic events, which is what you see here in this beagle dog here that has Lafor's disease. Burge and his group's done a lot of work in this space as well. The driving force of the disease are these sugar-like aggregates that are found in the cytoplasm from most tissues. These are called Laforabodies or polyglucosan bodies. And so Lafor's disease is a glycogen storage disease like adult polyglucosan body disease. So the really driving mechanism, which you heard from Sylvia for Lafor's disease, are mutations in either the gene that encodes Laforin or the gene that encodes malin. This results in aberrant glycogen metabolism, Laforabodies form, and many of us in the field, those at Indiana University, UT Southwestern, my lab, and in Spain have shown that those Laforabodies are what really drive the disease. And so as a collective community, there's been a number of mouse models that have been generated in Laforin knockout or malin knockout that recapitulate the disease to various degrees quite well. You see the Laforabodies here in the brains of these knockout animals. If we use an antibody to stain for the Laforabodies, what you see in the knockout animal, this is the Laforin knockout, it's just really every region of the brain is impacted. The cerebellum, the hippocampus, the cortex, both white and gray matter, all impacted by Laforabody accumulation. So the focus of the community is to inhibit the Laforabodies, which you heard from previous talk from Burge and Sylvia, or to degrade them, which is what you're going to hear from me, to impact and really modifies disease progression. So I just wanted to capture kind of where the Lafor disease community is at, and the GSD community as a whole. As Julie said, really exciting times where we've got antisense oligonucleotides that have gone through various models. I'll tell you today about the antibody enzyme fusions. You heard two different groups talking about small molecules. You're not going to, I don't think we'll hear about repurposing drugs or gene therapy, but there's some exciting progress on those fronts as well that's moving forward. So what I'll focus on are these antibody enzyme fusions, and these are really built on the back of a lot of success in the cancer field using antibody drug conjugates, where the antibody takes a specific compound to a certain cell type and delivers, in this case, a cytotoxic drug to destroy or kill the cancer cells. Well, Valerian Therapeutics initially pioneered this idea of utilizing a specific antibody that can cross the cell membrane. Valerian has been reincarnated as Parasail and Enable, so you'll see all three of these names scattered throughout my talk. Think of them loosely as one entity. Dustin Armstrong will be on in a few minutes and can answer some questions about that as needed. But what they found was this a fragment of an antibody, which is referred to as a Fab, that can penetrate into cells. And so the idea was that we could take an enzyme that could degrade Laforabodies and connect it to this Fab and penetrate into cells. And so we did this with Valerian and Enable. We made this fusion protein. They They generated a large quantity of it. It was quite pure. We tested in just in the test tube, if we took purified Lafora bodies, shown here from the mice, could this fusion protein degrade them? And the answer, you can see quite easily, is yes. Looking at under the microscope, that the Lafora bodies are degraded. And so the idea here is then that we could take this fusion protein, which is referred to as VAL-0417, it would gain entry into cells via this ENT2 transporter because of that Fab. There's a whole body of literature on how this works. And then once into the cell, the amylase portion of the fusion protein could degrade those Lafora bodies that are in the cytoplasm. And so this would be a way to remove those Lafora bodies. We've published on this work back in the end of 2019, so I'm not going to take you through all of that work, but just show you really the one key experiment from that paper. And so what we did was we took old Lafora disease mice. These mice show robust pathology, lots of Lafora bodies in the brain. We administered the drug continuously by intracerebroventricular administration for 7 days. And then at the end of those 7 days, we euthanized the mice and then looked at PAS staining, which you've heard a little bit about, that stains those polyglucosan bodies. And so this is what we saw. We were extremely pleased with this result. This on the far left here, you see the Lafora disease animals that are untreated. Here that I'm pointing at, these are Lafora disease animals that undergo the brain surgery for the ICV administration, and then they receive just vehicle for 7 days. And then the next panel, you've got the Lafora disease animals treated with VAL-0417. You see the Lafora bodies are gone and the tissue looks very reminiscent of a wild type untreated mouse. So we've completely ablated these Lafora bodies. And we've done this in every region of the brain. We were really stunned with the penetration and the biodistribution of the drug throughout really the entire parenchyma of the mouse brain. We could see that in the cerebral cortex, the thalamus, the cerebellum, the brainstem, hippocampus. Every region was really devoid of any Lafora bodies after that 7-day treatment. So that was super exciting, but the real question is, you've removed the Lafora bodies, does that have any impact on brain function? And the way that we answered that question was to go back and to think about the biochemistry that Or just talked about. You know, when glycogen is utilized, it's utilized to fuel central carbon metabolism. And central carbon metabolism is encompassed by all of these different pathways that back in our biochemistry courses, you have to memorize all of that, right? All of the metabolites from central carbon metabolism. So we wanted to ask, after treatment with the VAL-0417, is there any, are we rescuing any phenotype deficiency in central carbon metabolism? And so to do this, we utilized metabolomics. So to do these experiments, you take a piece of a mouse brain, you pulverize that into small particles. You can then extract those small metabolites out into a polar fraction and a pellet fraction, and then run those different fractions on a gas chromatography mass spectrometer. So in that polar fraction, you've got about 150 metabolites that are quite easy to quantify after derivatization and then analysis by GCMS. So you get these different peaks off of the GCMS. You can curate those and then do analyses of the metabolite profile from these animals. And so this is what we see. So each spot, each dot here on this graph is is one animal, and it's about 150 different metabolites, all concentrated into that one spot. So what this tells us, these data say, are that the Lafora disease animals have a different metabolic profile in the brain from wild type animals. This is just one way to look at the data, compress it down into a nice graph. Okay, so what happens if we treat the animals with PBS? We give these Lafora disease animals brain surgery. After brain surgery, obviously there's going to be inflammation. It's going to change your brain metabolism. So we do see a shift in brain metabolism, but it's still not normal like the wild type. But after we treat with VAL-0417 for 7 days, all 5 of the Lafora disease animals had an incredibly tight clustering of their brain metabolism, and it overlaps with the wild type animals. We can look at this by a different method. We can take the same data, plug it into an algorithm, and ask the algorithm to say which groups of these animals are most similar. the algorithm says, okay, here are all of your Lafora disease knockout animals that are untreated. They all look very similar and cluster together. And now here are the wild type animals and the Lafora disease animals treated with VAL 0417. And for these animals, the algorithm can't tell the difference between the wild type and the treated. So we've completely normalized now from this aspect, the brain metabolism of these animals. One additional test that we've recently published on in the last year is MALDI imaging. And the beauty of MALDI imaging is it starts to give you a spatial understanding of what's going on. In the previous data I showed you, we had to grind up the brain, and so you lose any spatial component. But with MALDI imaging, you're able to keep that spatial component. And so what we can do is take sections of a brain and then treat those sections with two different enzymes. Isoamylase is going to release the glucose chains so that we can analyze them on a mass spectrometer. At the same time, we treat the slide with this enzyme called pangase, and this pangase enzyme releases these sugar chains that decorate proteins called glycosylation. And so on the mass spectrometer, then we can differentiate the glucose polymers shown here versus the glycosylation polymers here in green. And so, you know, the key to these glycosylation decorations are that they're very critical for protein folding, for cell signaling, and for transporter function. And where else is transporter function more highlighted than in the brain, where you have to move around all of these neurotransmitters? And those are all highly glycosylated. So if we look at just one type of sugar decoration, this designated 1542 shown by this cartoon here of different sugars, this is what that decoration looks like in the wild type brain. The hot colors are high regions of where that type of glycosylation is at, and the blue colors are low regions. If we compare that to the Lafora disease animals, you see a massive difference between the wild type and the Lafora disease animals for this particular type of sugar decoration. Well, we can look at a number of different of these sugar decorations. Here's 3 different ones shown here. The pattern that you see in the wild type animals. And then if we look at what happens in the Laforan animals, you see that we have this hypoglycosylation defect in the Lafora disease animals. We now take those Lafora disease animals, we treat them with VAL 0417, and we see a rescue of this phenotype in every region of the brain for all of these different glycans that we've looked at. So I'll just close by talking about a little bit of unpublished data. There's also a second drug that Valerian has generated called VAL 1221. It has the same Fab, so the same antibody fragment that can gain access into cells via that ENT2 transporter, but it has the additional benefit that it's fused to the GAA enzyme. This is the enzyme that's mutated in Pompe disease, and it can access cells via the M6 phosphate, mannose-6 phosphate receptor. So now then you've got a fusion protein that has 2 different ways to access the cell cytoplasm. It can get into the cells via ENT2 and mannose-6 phosphate receptor. Additionally, this drug has already been in a clinical trial for Pompe disease. It was given by IV administration and you can learn more information about that clinical trial at this identifier here. There were no safety concerns from that clinical trial when the drug was given IV, and there were some benefits that were seen in very late stage Pompe disease patients. So we've tested VAL 1221 in the Laforan animals, and I'll just say that everything I've shown you for VAL 0417 today, we've also done the exact same experiments with VAL 1221. We see similarly exciting results with it, and we've got a paper that we're getting ready to submit on it. So I'll just close by sort of saying that we've tested these 2 fusion proteins in greater than 400 mice today in both the Laforan and the Malin knockout, as well as wild type animals. We've seen minimal to no safety concerns in the mice. The way to kind of think about these drugs is that, you know, the mother company and patents are held by Parasail. Enable, which you hear about, is more of an LD-centric company. We're working with both of them to move VAL 1221 on through the testing paradigm. And really the next big test that my lab is doing in collaboration with them is a canine test to look at safety in a large mammal, number one. Number two, to define the large mammal brain distribution, and then also the cell type penetration. So we're doing an initial 5 animal study. 3 will receive drug, 2 PBS. We'll collect CSF at day 14, and then at day 30, we'll complete that test and answer a lot of these questions here. So we think this is an exciting next step to get this drug into the clinic via ICV administration. Just want to thank the entire team that we work with, all of our funding from NIH, Cure Epilepsy, the Epilepsy Foundation, and really the outstanding collaborators that are driving this project. And I'll stop there. Thanks.
Thanks very much, Matthew. That's great. I think we will have a short presentation from Dustin Armstrong to sort of complement the sort of second part of the story.
Great. Hello, everyone.
Dustin is Dustin's from Paracel LLC. Pardon.
Hi, everyone. I appreciate the discussion from Matt leading into this. I'm Dustin Armstrong. I'm the founder and president of Paracel. As Matt mentioned, it's kind of the new reboot of Valerian, has control of all the IP and drug material. I was also the founder and CSO of Valerian, so I've been working with this technology for now a number of years. The key aspect that we have in this technology is that it allows for improved tissue targeting, so getting to the actual organ needed, whether it's the heart, brain, skeletal muscle. And then the second component is then once it reaches that organ, to be internalized into the actual cells that it needs to be delivered into. And it really achieves that, as Matt mentioned, via the antibody portion, in case of many of these fusions of Fab, and then the ENT2 transporter on these relative tissues. As Matt mentioned, our lead fusion is a FAB-GA fusion that was in a clinical trial for Pompe already, but now with Paracel, we're really trying to kind of have a renewed focus and look at indications where we can expand our clinical development into other polyglucosan diseases, where we can really take advantage of the unique mechanism of action of getting it to tissues needed, such as the brain and muscle, and then also degrading cytoplasmic aggregates, so internalizing into that particular intracellular compartment of cells. So just some background on why we have the rationale that we can get this enhanced targeting is that the ENT2 transporter has actually been shown to be upregulated in the cases of certain diseases, specifically in the brain and at the blood-brain barrier with neurological pathologies like stroke and epilepsy. And this was a recent 2021 paper where they found ENT2 elevation and also being active following different pathologic conditions. This new data actually highlights and kind of further solidifies earlier data with a research candidate that utilizes a similar antibody was shown to only penetrate to the brain in a stroke model when the stroke pathology was induced in rats. And so that was already seen that in a certain disease condition, it could penetrate to the brain greater. And now the rationale of ENT2 being upregulated is more established. And that's been further confirmed now by some independent findings in 2 other groups, where they've shown that other specific antibody variants also penetrate to the brain, in this case with tumor metastases. But they really here highlight in the paper that this presence of ENT2 on endothelial cells, such as the blood-brain barrier, and their upregulation with disease may allow for antibody targeting to that organ. And so that really is the key kind of target delivery aspect that this technology can bring. And I just wanted to highlight then the 2 in this relative like single diagram. And so to get to the brain, as we mentioned, there's the scenario where disease pathology may allow ENT2 activity to increase, and with systemic IV administration still get significant brain delivery. And so that's something that we're examining. But as Matt mentioned, a second alternative to bypass this would be to administer the drug through ICV or IV or IT delivery. And that's something that, as you said, we're collaborating with. We have some evidence in the mouse models. We're now looking at canine models. And what's great is the FAB-GA has certain properties that allow this to be formulated for that type of delivery. But as I mentioned, we already have clinical data now in Pompe with IV delivery, and with brain pathology, there's corresponding evidence that it may be sufficient for targeting alone because of this ENT2 upregulation. But just getting to the organ is not enough. It obviously has to be internalized into cells. And what's unique about our FAB-GAA fusion that Matt touched on is we have this FAB portion that can be internalized via the ENT2. GAA also has the GAA mannose-6-phosphate that can be internalized. But when that's internalized, it only goes specifically to lysosomes. And so the advantage of the ENT2 pathway is that we've seen delivery to the cytoplasm. And a lot of these polyglucosan diseases we discussed, the aggregated glycogen is actually in the cytoplasm. And as Matt pointed out, the fusion with the FAB amylase, the 417, is really great evidence for this ENT2 path of targeting because it lacks the GAA. Amylase alone doesn't penetrate cells and doesn't have that capability, but when fused with our FAB, as Matt showed, we got distribution throughout the brain with ICV delivery, and also within cells, it was able to degrade these aggregated LaFora bodies. And so that just, in this one slide kind of highlights, there's really two rationales and benefits from having our FAB fused to an enzyme. One is the delivery potential, and we've seen that even in healthy mice, we have improved delivery to certain organs that are really the key organs needed for delivery. And then secondly, to get into the cell to the right compartment, the FAB and ENT2-mediated pathway have that benefit of allowing that. And so just to kind of quickly summarize that the Val-1221 has these dual properties that allow for both enhanced delivery of the target organs, and that could be the case for CNS diseases, which is why we're, as Paracel, very anxious to look at now more CNS-focused indications. Val-1221 can uniquely act on cytoplasmic glycogen. And so we've seen this in a number of in vitro models where glycogen in the cytoplasm can be specifically degraded with our fusions. And now in animal models, we have Pompe data published. As Matt said, LaFora, we're about to be in submission, but we also have in vivo data in PRKG2 syndrome mice and also in GSD4 adult polyglucosan body disease mice. And so this preclinical package has now allowed us to have advancing clinical new designs and in discussions with the FDA. So because we have previous safety data with the Pompe, we currently have drug material that's released on hand, which is suitable for human administration. We've now also discussed with the FDA, we have an active IND that's been accepted for LaFora disease, which we're now actively pursuing into the clinic. And we've already had a pre-IND meeting where we discussed our profile, and the FDA found that we had sufficient data to support expanded development into a number of polyglucosan diseases, including adult polyglucosan body disease. So just to wrap up, Paracel obviously is a new reboot. It's a very small kind of boutique company at this stage. However, as I've talked about, we have very exciting technology, and most importantly, we have drug material and the clinical ready material to begin additional clinical studies. And so we're seeking that to both proof of concept, which has been discussed in terms of smaller patient numbers, but also advancing fully to pivotal studies. That is, as soon as next year, could have very meaningful data. And that's where you want to get to allow approval of drug and eventually insurance reimbursement and allow all patients of a disease to access that drug. So I appreciate your time and look forward to following up additional questions.
Thank you, Dustin. And may I take this opportunity to thank every speaker in this session. It's been really great innovation and also excitement and very inspiring. I'm conscious of time. It's around 12:25 your time, US East Coast time. And can I say, are the speakers all right to stay on for 5 to 10 minutes? I'm sure. But thank you very much for already answering some of the questions in the chat very diligently. I think if I can see a couple of questions in the chat, circling around the idea of translating work in diseases of LaFora or Pompe into relevance to this patient group. Yeah, maybe Julie and Matthew, maybe and Dustin, maybe that's a question I can sort of point to you first. I think Julie, this is a question asked by Lena. What does the future look like for other indications beyond Pompe that you show very nice work in terms of LaFora or APPD?
Yeah, thank you for the question, Wyatt. Obviously we're interested in trying to get this compound to as many patient populations that could benefit from it. Our focus needs to remain on Pompe at the moment. We need to understand pharmacokinetics, safety, tolerability, and then of course, efficacy. If this does work, then I think it potentially opens the door to investigating this directly in as many patient communities that could benefit. But just in terms of Burge mentioned how much clinical development costs, it's extremely challenging, and so we need to kind of take it one step at a time and really understand again that safety, that tolerability, and efficacy, and then go from there.
Yeah, thank you for this candid thought. That's great. Matthew and Dustin, in your presentation, of course, you alluded to also translating your approach to APBD. A similar question, but maybe in the direction of for your plans, do you think the preclinical work or the safety work that you have done will be considered similarly for APBD, or are there different considerations you need to move in this direction?
Yeah, I mean, I think Dustin's got, I think, spearheaded a great opportunity on the IV front. The drug, clearly there's peripheral issues in both diseases. And so administering the drug via IV makes a lot of sense. So, he and I have discussed him pushing that forward. We've got funding to do the ICV administration in the canines. And I think that's an important model to see, does the drug penetrate deep into the parenchyma of a large mammal, and is it safe in the brain of a large mammal? And so I think that tag teaming that makes a lot of sense towards pushing the drug forward. Dustin?
No, I think that's a good point. As I said, we do have some early data, whether it's ex vivo, just to show in these other glycogen storage diseases, one, can it degrade that type of species of glycogen? So we've kind of answered that in GSD4 models where isolated glycogen from different tissues is degraded ex vivo by our fusion. And then now in animal models, we do show proof of concept that we get, like Lafora, degradation of glycogen in various tissues from these mice. So the next really step then is clinically, as we mentioned, and that's really, I think, kind of a theme today, is identifying resources to then advance those into the clinic. And I think one advantage we have on the IV front is we have drug material, we have safety data, we have FDA support. So it's really a matter of matching, clinical investigators with the funding to support the logistics of the regulatory and administration needs of that trial. And so that's where we're actively kind of seeking investment from either grants, foundations, venture capitalists, or other partnering companies.
Thanks, Matthew. Thanks, Dustin. I have probably a question circling around combinatorial therapies or complementary approaches to ask a few people here. But meanwhile, if there are any other questions from the audience or from the speakers, please do put it into the comment box or raise your virtual hand or message me directly, and I can also point to the speakers. Maybe this is a question to Burge, but others also feel free to chip in also. To Julie's approach of preclinical, in the preclinical studies with ERT and SRT approach. Burge, do you see for your ASO and for the siRNA, you will also be considering in future development a combinatorial approach?
It may be needed. We just don't know yet. The ASO and also probably the small molecule, anything targeting like glycogen synthase, downregulating glycogen synthase activity, is likely going to stop progression of the disease. Sylvie has shown this formally with the ASO. You cannot make the existing bodies go away if you just stop glycogen synthase activity. So for an early onset patient, not early onset, someone who's recently diagnosed, where the symptoms are not yet severe, these treatments would be tantamount to a cure. It will stop progression of the disease. Since we're in an APBD meeting, this disease being relatively slowly progressive, if it's diagnosed early, treated early, then the patient does not go into the sick phase of the disease. But what about more advanced patients? We believe that removing the bodies the way Matthew and Dustin have been working and describing is kind of the way to go with that. So maybe a combination of removing the bodies and stopping their further formation would be one type of combination. And in fact, I wasn't aware, thank you, Julie, for sharing your results. That's kind of what Julie is doing. The ERT, you know, will remove the existing, and the SRT will prevent new formation.
Yeah, I would add to that. I think, you know, all of these glycogen storage disorders really within patients exist on a spectrum of severity and a spectrum of, you know, rate of disease progression. And I think the more we talk to all of the key clinicians, the more you realize how much this what does success look like for a patient will be very different relative to where they are, and what the therapeutic intervention is needed to really move the needle on quality of life could look very different. So I think, to me, that's what's so exciting about all of these talks, is that there's multiple different modalities, multiple different, you know, biological strategies, therapeutic strategies that could really have a huge impact on a whole host of these glycogen storage disorders, especially if they're considered sort of in combination over time as a tool for physicians. So,
thanks, Bert. Thanks, Julie. Matt, or any thoughts on sort of the approach of complementary combinatorial?
Yes, so first of all, I completely agree with Ber, Dr. Minassian, and I can also add that as our research unfolds, we find different targets that can be complementary. So some of them affect the synthesis, some of them affect the degradation. I presented the degradation, but of course, combination therapy is very promising if you have the targets. Okay, if you have the exact targets. So that's actually where we are heading. As a matter of fact, one thing that I wanted to mention is the importance of the pre-treatment. So we are, both APPD and LAFORA are neurodegenerative disorders. A dead neuron is a dead neuron. So in our case, it was actually even extreme. Only the preventive treatment worked. What I showed was preventive treatment. If you apply the treatment after a disease onset, so around the age of 6 months in the case of APPD, it wasn't effective. So this is very important to realize. And this is just texted Julie, I think, or you, Wyatt. Some irreversible damage, for instance, also occurs in Pompe. And according to the literature, even ERT treatment, which starts just at birth, many times has a limited efficacy because there, even in pregnancy, there are some damages which are unfortunately irreversible. So the timing is really something that we have to be attentive to.
Just one quick comment, Wyatt. Julie's experiment, I think, is fantastic for this discussion. Just point out that the ERT Lumazyme, that's the same enzyme that Val-1221 is based on. It's the GAA enzyme. So, a lot of similarities here with regarding proof of concept, and all the concepts are triangulating to tell us the same answer, which I think is fantastic.
Great point. Thanks, Matthew. Thanks all. I think in the message box there are a few emerging questions. If I haven't missed, Cheryl, you have a question for Birch. Slightly polyglucosan bodies have accumulated already in when APPD patients are diagnosed, and it will be sort of compensated for years before it manifests. Probably that's a comment for you.
Yes. As Matthew said, the body of evidence in all these diseases is pretty convincing that the polyglucosan bodies lead to the downstream impacts, in part through neuroinflammation and then neurodegeneration, whether it's direct neurodegeneration or due to this very active neuroinflammation in the brain. And the whole thing is progressive. So yeah, I mean, the earlier you intervene, the better is the answer. Like pretty much any disease, actually definitely progressive diseases.
Thanks, Birch. And Cheryl has a question for Dustin, I think. Would you be able to share your learnings or experience or information on FDA's readiness to look at several glycogen storage diseases together in your clinical studies?
Yeah, so as I mentioned, we've had discussions with the FDA in a pre-IND meeting where we discussed our preclinical package as well as our clinical data from Pompe, and informed them that we think this could have applicability and to expand clinical trials to other polyglucosan diseases that all have the same etiology, that there's a cytoplasmic elevation of glycogen, and they were very receptive to that. Obviously, for different diseases, you can actually in a single trial have different endpoints, and so that's one advantage where there's some differences in diseases, either what target organs or what you'd want to look for as crucial endpoints. And so there allows some commonality, but also different. And so you could envision a pivotal trial where you take, a certain number of Lafora patients, adult polyglucosan body patients, and others, and actually have it in a singular trial as one option, or you could separate them and go to different divisions of the FDA and do it more independently. And so from our standpoint, we're evaluating financial logistical operations that would do that. But I think we're kind of at a crucial point now where, if there is the ability to add patients to Lafora trials as we're starting, there would be some significant cost saving advantages if you could utilize similar clinical sites, or if you go through the regulatory process in different countries already to kind of piggyback them. And so that's something that, we're speaking with different investors. Some, unfortunately, maybe have different diseases that they're willing to invest in than others. And so trying to, match that in terms of investment, but also best clinical need and availability is, I think, why we're talking to groups like yourselves and trying to understand, is there ability to develop some of these more in parallel and not, individually.
Thanks, Dustin. On the point of sort of trial and downstream development, I think there's a question for Or and for Laszlo's work on sort of thinking about what would be needed as the next step to start a trial as soon as possible for your product.
I'm sorry, can you just repeat that? I didn't know if that was.
I think that's a question for Or. Sorry.
Okay.
Sorry. Yeah. So the question, I think, is regarding Dr. and Laszlo's work. What is needed to start?
Yeah, so okay,
possible for your
product. So I missed it. So yes, I think it was actually Dr. Laszlo already referred to this question. So I'll just repeat. At the moment, we are in the midst of compassionate treatment. Once this is completed, approximately, let's say in half a year, something like this, then we have the critical mass to continue with the clinical trial, which obviously, as Dr. Minassian said, it depends on funding, and that's why we have the new company and representatives are here, and we are recruiting funds for the clinical trial. So first we have to go to the compassionate treatment, and so it's finished.
I'm sorry, this is Cheryl. Am I able to, thank you. I apologize, or I don't mean to cut you off. I just want to add to what you've said. Thank you so much. So my name is Cheryl Fitzeratas, and I'm working with GHF. I just wanted to clarify, this is a very unusual situation where this compound A, as it's called, has already gone into 2 patients under this urgent compassionate use program that's very particular, very specific to the Israeli health system, right? So that's not something we would see happening in most countries. So it's very exciting to have that opportunity. At the same time, of course, for regulatory approval, this compound, as just like any small molecule, would have to go through the rigorous safety and toxicology, CMC programs, et cetera. So we are doing those. The company is also looking for investments in order to move those processes along simultaneously to the work that Professor Lossos explained and shared with us today. So yes, he is of course, from a clinical perspective, collecting data and looking to see if there's any safety signals, et cetera. And we would hope to expand the clinical program with Professor Losas, as well as other, of course, researchers around the globe. So just like other companies that are here, this is an orphan disease. We are looking for investments as well, and hope to move the program along as rapidly as we can, given that it's already been in the clinic, right? So it's really sort of flipped around, and we thank the family that established GHF for really putting their efforts to make that happen. So thank you for letting me share.
Thanks a lot, Jennifer. Yeah, thanks for sharing your thought and your own experience in this and the insight in this. So that's great. Calliope, are we going through most of the questions already? I'm conscious of time.
We still have a few of them, but
Okay.
we are a little bit over time, but we'll give everyone a 15-minute break before the next thing starts.
So 4 more minutes, okay. Oh,
Okay. I just wanted to raise a question to the entire panel. Has anyone looked at glycogen in exosomes or induction of exocytosis as a way of clearing polyglucosans of glycogen? Some years ago, I did observe polyglucosans in exosomes, but I didn't follow this. So it was just a hint. I was wondering if anyone from the panel has, number one, looked at polyglucosans in exosomes, or even try to induce exocytosis as a strategy for clearing polyglucosans.
so if I understand, you're asking panel here whether people have
If anyone has considered that or looked at polyglucosans in exosomes.
So we haven't, but there is a body of literature in Pompeii from Nina Rabin's group at NIH that has demonstrated in cell culture models lysosomal or multivesicular body exocytosis as a clearance mechanism that's GAA independent. And that's sort of what we are interpreting right now as the mechanism for that slow clearance that does occur in animals. Is perhaps in muscles in particular, there's, a stronger pathway for that, or, there could be tissue specific differences, but, we're not.
Type 1, type 2,
Say again?
Type 2, which muscles,
Oh, I'm not sure.
Julie, the glycogen accumulations in Pompeii, couldn't it just be phosphorylase? Could they just leak out into the cytosol and be digested?
It could be. I think that the consensus from the literature that I've come to is that the lysosomal membrane integrity breakdown doesn't really happen until the muscle cells are absolutely chocked full with these organelles. And then you just have sort of massive simultaneous collapse. So it's harder for me to imagine that being a constant kind of pathway for turnover. I'm also, by training sort of a lysosomal biologist, so I fancy the idea of lysosomal exocytosis, but I think the data, especially in animals, is really not there to know conclusively whether or not that's a significant pathway.
Thank you.
Thanks. And I think Matthew put a paper link there on phagocytosis studies in brain. Any other questions or any lasting comments? If not, then I think we had a good discussion. We had sort of very excellent summaries from really different stages of the work, and I think we're keen to keep progress to hear about the progress in some of these work. And lastly, to thank everybody for speaking here and for contributing and for being in this session. Thank you very much.